All right, guys, let's talk about photosynthesis and cellular respiration. This can be challenging for some students, but there's a couple things if you keep in mind, it will make your life much easier. Keep in mind that the entire goal of this whole thing is that you are going to be able to describe the flow of energy from the sun into sugars, and then how organisms then manipulate that energy to help them maintain homeostasis. Okay, that's the whole point. There's two ways of doing this. The first thing that we have to keep in mind is that there's a process called photosynthesis, and the goal of photosynthesis is to trap energy so we can use it later. Okay, then there's another process called respiration, and the goal of that process is to release that energy. And in fact, there's two different organelles that do this, but we're going to dive into it, okay? But just remember that the goal of photosynthesis is to trap energy. The goal of respiration is to release energy. Next slide. There we go. So let's talk about photosynthesis, which again is the process of trapping or containing energy, okay? The formula looks kind of long, right? But the challenge of all of this is that, or what we're shooting for during photosynthesis, is we're trying to make this molecule. This is how we trap energy. This is called a sugar. Specifically, this is glucose. And it's not writing for me right there. Come on. Come on. Nope, it's not going to write for me. Whatever. This is sugar, okay? Sugar is just basically a battery. Okay, we need to make this, and a plant can just make it if it has the right resources. And the reason why it's doing that, it can actually get access to energy through the sunlight. It doesn't need the sugar during the day. The problem is that there are other times of the year, right? We don't have just days, we have nights. So in order to have energy to survive, to maintain homeostasis, we need energy for that time as well, right? During nighttime, there is no sunlight. So what plants have to do is store that energy for later, okay? In order to make a sugar molecule, we need a couple things. We need specifically carbon, hydrogen, and oxygen. The plant gets the carbon and the hydrogen from two different sources, right? It gets the carbon from carbon dioxide, and it gets the hydrogen from water. And it ends up, because of it needs 6, right? It needs 6 carbons and it needs 12 hydrogens. It gets that enough from there, but it also needs 6 oxygens, but we end up with actually a massive amount of oxygen. We end up with 12 oxygens from the carbon dioxide and another 6 from here. And all we really need is this 6. So we have 12 that we have to get rid of. And that's why we have this over here, is we're just getting rid of the waste product. But again, the whole point is to make a sugar, to make a high energy molecule. So I prefer, when I draw and talk about photosynthesis, to draw it like this instead, where I have a line going across the middle. Above the line I can draw high energy molecules, and below the line I can draw low energy molecules. So some low energy molecules that are involved in this process, right, is we have 6 carbon dioxide molecules, because we need that carbon. And we also need 6 water molecules, because we need 12, that's H2O, we need 12 hydrogens to make this whole thing work, okay? So if I have access to those materials, I can make a high energy molecule, but I'm lacking something. I'm missing something very crucially important. That very important, crucial thing, right, is I need some source of energy. And in photosynthesis, we're talking about the sun. The sun is constantly radiating energy. So plants can capture that energy using the raw materials of these on the left, which is what we call the reactants of a chemical formula. And we end up with a high energy molecule called sugar, C6H12O6, right? We now have this high energy molecule, okay? We also end up with a bunch of extra materials that don't have high energy, right? It's all this extra oxygen. So we end up with specifically 6 of these oxygen molecules. But what I like about doing this way is it really emphasizes that the whole goal is that we end up with these high energy molecules that we can use at night. Okay, so that is photosynthesis. Photosynthesis happens specifically in a specialized organelle called a chloroplast. That chloroplast is responsible for doing this process that we call photosynthesis. It's responsible for storing energy in sugar molecules. If we look at staining of a cell, all these little green dots everywhere are called chloroplasts. If we look a little bit closer, we can see the structure inside that enables it to do this. It's a machine that can capture sunlight energy and then pack other things together to trap that energy, right? So we have all these different weird little structures inside the chloroplast to let it do that. So let's do some practice problems. First, we need to make a little note that if you add CO2 to water, you get something that is more acidic. In case you have forgotten, acidic is a remark on the pH of a substance. 7 is something that's neutral. If we move away from 7 in either way, we're going to change the pH to something that's not neutral. If we move up on the scale, we're making something that is basic. If we move down on the scale, we are getting more acidic. So as I just mentioned, if you add CO2 to something, to water specifically, it makes it more acidic. So it would make it go this way if I added CO2 to a liquid. So now that we know that, let's look at a practice problem here. We have a compound called bromothymol blue, which changes color in different pH substances. If you look closely at this over on the side right, where it says blank, there's nothing in that one. But if we have a solution that's really close to neutral, 7.2, bromothymol blue is kind of this teal color. But as we move this way and things get more and more acidic, it gets more and more yellow. And as we move this way and things get more and more basic, right, we get blue. Okay, so if we look at the setup here, right here we have a plant, right here we have a snail, and right here we have both a plant and a snail. Okay, same setup underneath in this area right here, right? Same exact setup. So in this one, right here, it's exposed to sunlight. That's a great sun. In this one, it is not. It is not exposed to any kind of sunlight at all. Okay, so let's look at those results. Let's look at what happens. Let me go ahead and erase all this crap. There we go. I'm going to go ahead and move. Maybe I can't move that. All right. So let's look at some questions and let's look at the implications of all this. Notice that the vial with the snail in it has turned yellow. Let's do a laser pointer here, right? This one right here has turned yellow. It has a snail in it, okay? So in other words, it is not doing photosynthesis. But this one that just has a plant in it stays blue, which means basic, not acidic, right? And this one with both a snail and a plant has stayed blue. In this example, where there's no sunlight, just as a reminder, right, this one right here has no access to light at all, okay? Notice that all the tubes turned yellow when left in the dark. So if we were to describe what is happening in both the light and dark experimental groups, here's what I notice. I notice that when we look at this top right-hand corner, the one that was exposed to the light, that any tube with a snail, or excuse me, not with the snail, any tube with a plant stayed basic. Some wonderful handwriting there. And then I noticed the one with the snail had a decrease in pH. It became much more acidic. And then if I look at the darkness ones, right, and the dark ones, all tubes became acidic. Which is fascinating, okay? So even though what this basically is saying, right, to me, is that the tube in the sunlight, there's an animal in there that's not doing photosynthesis, whereas if you're doing photosynthesis, you're pulling carbon dioxide out of the water, so it's going to make it more and more basic, right, if you have a plant doing photosynthesis. So I noticed that in each of the tubes in the sunlight where the plant is able to do photosynthesis, it's pulling carbon dioxide out of the water so fast that it stays basic, even when there's an animal that's adding carbon dioxide to it. When I look at test tube D4, right here, up here at the top, that explains to me why that's happening, is that the plant's ability to do photosynthesis and pull carbon dioxide out of the water is even more rapid than that of the snail. Let's do another practice problem. In a lab investigation designed to demonstrate one aspect of photosynthesis, 3 test tubes were treated as shown above. The test tubes were then placed under a bright lamp for 24 hours. Which of the following is the best explanation for the observed color change in tube number 2? So it's asking about tube number 2 right there. So I'm going to make a couple quick notes to myself. I noticed that the initial color is blue, which means that the pH is low in that first one, right? That's a low pH if it's blue. So both in number 1, where there's a plant and the solution, it always stays at a low pH. In this one, where there's a plant and there's the carbonated water and the bromothymol, over time it turns blue. So it goes from a high pH to a low pH, which the plant is involved in. And that makes sense because it's pulling, scratch that, reverse that. It goes from a low pH to a high pH, which makes sense because the plant is pulling carbon dioxide out of the solution, which makes it not be acidic, that is to say basic, right? And so we're getting a higher and higher pH. In this one, where there is no plant, it's yellow and it stays yellow, which makes sense, right? This is a high pH. Oops, and I had this one back. How to make this confusing. So this is now a low pH, and it just stays as a low pH. So we're asking about tube number 2, and it wants to know what's happening for the observed color change in tube 2. Clearly, there's an aquatic plant in here that is using up carbon dioxide, which is then raising the pH of the solution. So we're going to go with option C. Okay, let's do one more. The green aquatic plant represented in the diagram below was exposed to light for several hours. Which gas would most likely be found in the greatest amount in the bubbles? Well, I know that during photosynthesis, there are reactants, right? This is the formula. We have reactants there and products here. The products of photosynthesis are going to be the sugar, C6H12O6, which is not letting me write there for whatever reason, the sugar. And the other one is oxygen. So since I know it's making as a product oxygen, I'm going to go ahead and say oxygen. Okay, so let's talk about cellular respiration real quick. In cellular respiration, the whole point of cellular respiration was to take that sugar that we made in photosynthesis and to break it apart so we can now have access to the energy that was in it. The entire point of all of that, right, is just to produce and release the energy that is stored in there. So if I was to rewrite the formula that we talked about in photosynthesis, but let's now do respiration, right, I have a high energy molecule, C6H12O6, plus a low energy molecule known as oxygen, right? Let's rewrite that to be less confusing. Let's see this here, let's erase this. Let's rewrite that. We're going to take our low energy molecule, oxygen, plus our high energy molecule, C6H12O6. And the point is to release that energy so that our mitochondria can use it to make ATP, right? Which is a separate process. But we now end up with these low energy molecules of CO2 and H2O. Come on, let me write. There we go. Okay, so that's the whole point of respiration, right? Is this step right here. Release the energy that was trapped in the sugar during photosynthesis so that we can do things with it. In this case, make ATP. Where does this happen? This happens in a specialized organelle called a mitochondria. And the mitochondria is responsible for releasing the energy in the sugar, right? Plants have these mitochondria to release at nighttime the energy that they stored during the daytime. Animals can't do photosynthesis, but we can steal sugars from plants and use the same exact process in our mitochondria to release that energy. So mitochondria, if we look at a cell stain, are all these little red stained things, right? There's clouds of them in different cells, many of them. And if we look closely at them, we have this structure right here, which is the mitochondria. It's going to take in sugar, and it's going to release as waste products the broken apart sugar, right? The reactants, or excuse me, the products of doing this is going to be water. Keeps blanking on me and not letting me write. It's going to be water, and it's also going to be CO2. But the reason it's doing that is trying to extract the energy from the sugar, right? And it can use that energy to then make this little compound known as ATP. Okay. So let's do some practice problems. Let's see how well we understand respiration. Here's a graph. Shows over time the concentration of oxygen and CO2. Oxygen is being this red line here, carbon dioxide being this blue line here. It's showing how much of it there is, right? Lots of it or none of it, and this is over time. So if we look at that graph, we can ask ourselves a couple of questions. What's happening in the graph? Well, we notice that over time, over the course of a day specifically, because it's 24 hours, the CO2 levels are rising quite a bit, and the O2 levels are dropping quite a bit. So why would the CO2 be increasing? Well, the answer is because cellular respiration is happening, which has as one of its products CO2. What would happen if we put a plant into the tube with the animal and did the experiment again? Why would this be different than the current experiment? Notice that in this case, we just have an animal, but if we put a plant in there, there would be some regulation. It could use that carbon dioxide and drive that down, and then the animal would be able to cycle back and forth. If we kept the test tube with the plant and the organism in the dark, we would actually see a greater increase in carbon dioxide more quickly, as both the plant and the animal are doing that, and a quicker drop of CO2, of oxygen, because again, they're both using it at that point. So which of the following are products of cellular respiration? We are ending up with both, let's see here, which option? Carbon dioxide, and we're releasing a ton of energy. So I would go with option A. Which is one of the reactants for cellular respiration? Well, we need sugar, and we also need oxygen to make it work well, but that's not an option, so we would just go with A, glucose. So the objectives were describe how a cellular respiration releases energy from sugar molecules that can be used to drive other chemical reactions in an organism, right? We're focusing on the releasing here. In objective 2, describe how photosynthesis converts light energy into chemical energy. We're trying to talk about that storage process of how it converts that energy. For objective 3, we're looking to describe how the molecules use and how energy transfers in the photosynthesis and cellular respiration. That's been our focus the whole time. How energy is trapped in a sugar and then released is the process of photosynthesis and respiration. So I hope that you found that helpful. We'll do another video later on. These are just a bunch of practice problems that we don't have time to talk about right now, but we can go into a much more advanced version. If you're interested, come approach me and we'll chat about it. Thanks. See ya.